Thank you for that generous introduction. You left out the thing that I'm most proud of, which is in the summers I go to Tibetan monasteries and I teach neuroscience to Tibetan monks in India. And that's a really interesting thing to do. So I'm very happy to be here, and I'm going to talk about our most recent work. I moved to University of Oregon about 2 years ago, a year and a half. And so moving, I'm like starting over. I'm like an assistant professor because I came with one undergraduate and we built a whole new lab and hired all new people. And we've been training them in mouse colony and everything. So I've only have modest productivity in the last year and a half, but hopefully in the future we'll be able to make good use of that move. I moved to the West Coast because we came from the West Coast and we always wanted to go back, and Oregon is a beautiful state. So these are the people that were at Yale that contributed to the work I'm going to talk about. Specifically, this guy right here, Matt McGinley, he did the work that I'm going to focus on quite a bit, and Garrett Nesky also did quite a bit of the work in the middle. This is my new lab at the University of Oregon. This is the student that came with me and helped set everything up. He's going off to medical school now. And these are my new postdocs and grad students. University of Oregon is undergoing a renovation. Most of the campus has been renovated or is being renovated. And it's in large part because of one student. His name was Phil Knight, and he was obsessed with shoes. And when he graduated, he went to a Japanese company and convinced them that he owned a shoe company, which he didn't. And they gave him some Tiger shoes, and he came back and he started a shoe company called Blue Ribbon Shoes, and he sold the shoes out of the trunk of his car at track meets, and then got the Japanese company to keep feeding him shoes. And he kept selling them, and then now he's worth $30 billion because he owns Nike. And so Phil Knight has given many very generous donations, and now he's turned his attention to science, and he gave $1 billion to University of Oregon and OHSU to build a new science building that's here. And this is going to be partly neuroscience, and this bridge here comes over to our building, which is currently neuroscience. We have two buildings. This is one of them for the Institute of Neuroscience. And in the 90s, when the internet was young, I actually bought neurobiology.org, so now it points to University of Oregon. So, so I'm going to talk about science that we're looking at now. And for the longest time, I was interested in sleep and waking. And this is my grandson, who was born 2 years ago. His birthday's in a day, I think. When he was young, when he was newborn, going to sleep and awake. And there's a major transition in the brain, right? It's probably the biggest one that a normal person has. And we were able, with many other labs, including Steryad's lab here in Canada at Laval University, to figure out the cellular mechanisms and the network mechanisms of sleep rhythms and why they go away with waking. But then I started looking into, or other labs started looking into waking state and started noticing rapid variations in state. And those rapid variations in state determine ability to perform. So if a high performance athlete is doing a repetitive task, what is it that allows them to hit the bullseye or not, so to speak? There's variations in the brain that are going on all the time. I call it seminar behavior. You know, you're paying attention right now, but I guarantee you in 10 minutes, some of you are not going to be paying attention, or maybe phasing out, and then you'll come back and you'll go in and out. These rapid variations are kind of like the big variations of sleep and waking, and they determine your ability to perform. So let's look real quick. By the way, if there's any questions, you know, feel free to interrupt me because I'd like to have a discussion. Let's look at the EEG, the classical view of the EEG. In the human, when you're deep asleep, you have slow rhythms in your EEG. And we now know from Sterod's work and my own work that these are up and down states. The network is on and off, on and off, on and off. And it does this in a half a hertz to 4 hertz rhythm, okay? And that's characteristic of deep sleep. If you're in a lighter stage of sleep, you have spindle waves, which is highlighted there in pink, and those come every few seconds, and we know the mechanisms of those. They're thalamic cortical rhythm. But if you're awake, those rhythms, the traditional story is those rhythms are suppressed, and now you're having a lot of gamma waves, and your brain is active, and you're doing processing. Okay, the slow waves were thought to be kind of anti-processing, and the higher frequency rhythms or the suppression of slow waves would be pro-processing, pro-cognition. Keep that in mind because that's going to be important for understanding this graph, which is taught as a basic psychology concept. And it was discovered over 100 years ago by Yerkes and Dodson. It's called the Yerkes-Dodson curve. And they did a simple experiment with rats and found that if the rat was very stressed, the performance on the task was low. And if the rat was drowsy or inattentive, the performance was low. And the performance peaked at some intermediate level of stress or arousal. So they hypothesized an inverted U of performance versus stress or arousal. All right? And we all know this from kind of practical experience. You know, you have to drink just the right amount of coffee, otherwise you're going to be too wired or not awake enough to perform on the exam or the whatever you're trying to do. So when we moved from slices to in vivo work in waking mice, we wanted to know what the state of the mouse was because the recordings we were seeing from people in the field was suggestive to me that the animals were either drowsy or asleep, or I didn't know how attentive they were. So Matt McGinley was an auditory physiologist. So he said, okay, well, I'll design an auditory task. Actually, he didn't design it. It was already in use in the field. In this task, the animal hears complex sounds. It's right here. And they sound kind of like ray guns, you know, like this. And then if there was a pure tone in that complex sound, the animal was cued then, he could lick and get a reward, all right? And it's a head-fixed mouse on a wheel that you walk on. And with the head fixation, we could do recordings or imaging. So the tone could be very loud or it could be very light. We varied it randomly, all right? So they're motivated to do this because this is when they get their water. They're water restricted. So here's a movie of a mouse doing the task. That's the lick port right there. And now watch this pupil. We took these movies just to, you know, be able to look at what the mouse is doing. Now we back the movie up and we get about the front half of the mouse. But the pupil diameter turned out to tell us a lot about the rapid state changes. This movie is in real time. He's sitting there. Now he's waking up. His pupil was really small. He was really kind of drowsy. And now you see it getting small again. And now you see it getting larger. And you can see the whiskers twitches too. All right, he's kind of relaxed. He's not doing much. His pupil is getting smaller. Now he's perking up. He's whisking and he's licking. And now his pupil is getting quite large. He's almost stressed at this point or very excited. And now he's starting to calm down, and then he gets a little excited again. Those rapid variations, and that's in real time, in the pupil diameter are mirrored by equally large and rapid variations in the brain state of the mouse. And I'll show you the evidence for that in a second. So that's how fast your brain state is changing. In fact, you know, when I was younger, everybody talked about how unreliable the brain was, and so we must, you know, average a lot to get a reliable signal. The major, now he's almost asleep, but he's waking back up again. the major factor for this variation and trial-to-trial variation in the brain is state. And if you take account the state of the animal and the fact that it changes so quickly, then the brain seems much more like a machine that does things very accurately. You know, think about it. How can if the brain was that noisy, how would it be possible for an archer to hit the bullseye many times in a row? So, you know, I went, after we found this, I went on YouTube and searched pupil diameter, and there's a whole lot of videos of people's pets, especially cats, because nocturnal animals have exaggerated pupil diameter changes. You know, cats nocturnal, mice are nocturnal. They need to be able to see in the dark. So I want to show you this video. Have any of you seen this video? Okay, this is a cat. Wait a second. Let's see. I have sound. Oh, that's good. Okay, let's try it here. Go forward, play. Nope, sound's not working. You can hear it here. It's watching a horror movie. You can see the pupil diameter changes. All right, so those pupil diameter changes went with the amount of attention the cat was, and fear that the cat was experiencing, it seems. Same in people, except the pupil diameter changes in us are very small because we're diurnal animals. So we were recording the hippocampus and cortex EEG to try and figure out the state of the animal. And when we compared pupil diameter to those things, we found some really unbelievable correlations. So here's an example of one of those. So this is what are called sharp wave ripples in the hippocampus. This is a sharp wave, and that's a ripple. These tend to occur when the mouse is, we were always told that when the mouse is sitting still, they occur, and then when the mouse runs, you get theta. As if the state of the brain is one or the other. It's almost like 0 or 1. But that's actually far from the truth. It's a continuum. So let me show you this example of sharp wave ripple rate versus pupil diameter versus walking. So up is fewer ripples, and up is dilation. So up would be arousal, and you get fewer ripples with. And if you plot on time, this is 20 seconds, the ripple rate is constantly changing like this, and it goes exactly with pupil diameter in red. And here the animal decided to walk for a little bit, and you see the pupils dilate quite a bit. The animal gets rather aroused. He gets aroused, then he walks, and then he stops walking, and then he relaxes. And the pupil rate decreases at the same time. But then over here, the animal is just sitting there. He's not walking. And you see there are periods where the animal is aroused. The pupil rate, the ripple rate is low. But then there are a few seconds where it's the opposite. And then there's 20 seconds of one, and then a few seconds of another, and then 20 seconds of another. And it's a continuum back and forth like this in the mouse. And this causes a huge amount of variability in the data. Then, even more unbelievable, we did wholesale recordings in the cortex, auditory cortex of the mouse, and we found that the membrane potential of the neurons was highly correlated with pupil diameter in some neurons. So here's the membrane potential of this neuron, and it's going along, and you see these bumps of electrical activity. and then it comes down and there's these rhythms. This is the slow rhythm. And then there's a couple of bumps of electrical activity, and then there's this slow rhythm. And then there's a depolarization. These 4 or 5 depolarizations all associated with pupil dilations. And that's because they're associated with increase in attentiveness or arousal. We call this microarousal because it only lasts for 2 or 3 seconds. All right? So the animal was quite relaxed and he had slow rhythms in his cortex going on. This is synaptic activity, by the way. But when he had these microarousals, the cortex kind of went into a new state and then went back. All right? Let me show you what happens now when the animal walks. When the animal decides to go for a walk, what happens first is the membrane potential of the cortex depolarizes, the pupil dilates, and then the animal walks. If the animal stops walking, the opposite happens. The pupil goes back to normal slowly, and the membrane potential of the cell comes back down. And then you have these micro arousals occurring spontaneously. Okay? If you took a filter and filtered this to very low frequencies and overlaid it, it would look just like pupil diameter. Now, so at University of Oregon, we've set up a lot of different newer techniques to try and examine the neural mechanisms of this state modulation that's going on. And one of those is wide field. So we have wide field microscopes looking down on the mouse cortex through the skull. And GCAMP6 is so bright that you can see the brain right through the skull and you can see neural activity. So this is neural activity evoked by sound. That's the auditory cortex in red. This is neural activity evoked by visual inputs. That's in red back there. And that dark brown spot there is whisking. The whisker stimulus was making a sound, so it activated both the somatosensory and the auditory cortex. And then if you look at this activity in the mouse that's sitting there, it looks like this. This is sped up 2 times. So the mouse's brain is just constantly in flux. This is mainly somatosensory cortex here, motor up here, visual back here, and auditory down here. So you can see mostly somatosensory areas are constantly in flux. All right? And I'm going to show you in a little bit why that is. In fact, I'm going to point it out here. If you look at the pupil diameter and the whiskering of the mouse, the whisker movements, and the walking of the awake mouse, you see that the whiskers are often moving. They're moving in twitches like this, just small twitches. And then the animal will purposefully whisk. And those purposeful whisks are typically associated with large pupil dilations and often with walking. Okay? Now, we also built a mesoscope. A mesoscope, let me see. Let's go back. A mesoscope allows you to see a 5 millimeter by 5 millimeter view of the, this is the objective with his thumb. It's about the size of a Coke can. And the bottom of it is about the size of a mouse's head. And you replace the skull of the mouse with glass, with a crystal skull. It's called a curved piece of glass like this. And then you can image 5 millimeter by 5 millimeter view at a cellular resolution with a 2-photon microscope. So it's kind of like an amazingly precise fMRI, if you will, of the cortex. So, but at the whole 5 millimeter by 5 millimeter, there's only about 4 pixels per cell body. So the resolution at the cell body is pretty low. So what we typically do is you pick out random regions, and they can be anywhere you want, X, Y, and Z. And the microscope can jump in a couple of milliseconds between each random region. So you could say, well, I want to monitor somatosensory cortex, auditory cortex, visual cortex, and at the same time. And that's what those are on the right. So, sorry. I have to go back. Go forward. There. You're watching the pupil, you're watching the mouse, you're watching the neurons, all at the same time. And that's where we are right now. And I'm going to talk about that data in just a second. So here's an example. So you're watching like motor cortex, somatosensory cortex, visual cortex, and the activity in the GCAM6. And then you see these flashes. You're just sitting there every once in a while. It's kind of random. It's kind of doing your own thing. But there's a lot of correlated activity even in these broad regions. All right? So while we were setting this up, Carandini's lab, Stringer, who's the first author, she's at Janelia now. They put out a paper in bioRxiv, and I understand it's in press now, where they did a very similar thing. And this is very current, I think. This is how people in the mouse field are thinking about this. They imaged about 10,000 neurons of the visual cortex. They didn't have a mesoscope, they just had the local region. And they imaged the mouse. So the job now for mouse physiologists is to compare the two images. How much of the brain activity explains the behavior and how much of the behavior explains the brain, all right? And so it's a lot of analysis of images. And there's a lot of discussion about how to analyze those images, especially the behavioral ones, or both actually. The way they analyzed the image was to look at the principal components of the video of the face. And they found that they could explain about 25% of the variance of the neural activity by just looking at the first 16 or so principal components. And the most important principal components was simply movements of the whiskers. So here we have running, and here we have pupil diameter, and here we have whisking. And here the neurons are segregated into likeness. So that if the more like a neuron is to another neuron, the closer it is in this graph. So you could see that some neurons are very strongly modulated by the movements of the whisker. Okay? So here is, that's running and whisking. These neurons are very modulated by running. Some of them are very modulated by whisking, and those things are all correlated. You see pupil diameter, whisking, and running are all correlated with each other, but not always going together. Here's the animal's whisking, but not running. All right? And they could explain a significant amount of the variance in this 10,000 neurons by just watching the mouse. Indeed, when we did whole cell recordings in a waking mouse, this is just spontaneous now. This is work by a postdoc named Dennis Nessvogel in my lab at Oregon. He looked at pupil diameter, whisker movements, and running, and membrane potential and action potential of a layer 4, layer 5 neuron in visual cortex of the mouse. And what you find is that if you overlay, and I'm sorry this is a little hard to see in the back, but the green, which is whisker movements, and the black, which is membrane potential, down here, these whisker twitches or whisker movements like this all correspond to membrane potential depolarization in the visual cortical neuron. And here the animal is whisking quite purposely, very large whisker movements. And here he is whisking and running. And then the membrane potential and the whisking went together very nicely and the pupil dilated. All right? This is 5 seconds. So this is a transition from one state to another within much less than a second, within 100 milliseconds or so. It lasted for maybe 15 or 20 seconds and then went back. And then he transitioned for maybe 1 or 2 seconds and then went back, and then these little 1 or 2 second bouts. And they have a big effect on the membrane potential of the cell. That's just more of the same with neural pixel probes instead of calcium imaging. Also from Carandini lab. So the point is that the brain is reflecting behavior and the behavior is reflecting the brain. The mouse is doing a lot of different things with his face. You know, if you watch the videos, just watch them, you can see they have very exquisite control of their nose. They're doing interesting things with their nostrils and their nose, their lips, their whiskers, their eyes. And when a mouse actually is running around, they move their eyes a lot to keep things centered. In HitFix, they don't move them that much. But they do a lot of different things, and that's explaining a lot of the variability that you get in neural data, all right? So we looked at just one of those things, which is what we call arousal, and we equate arousal with pupil dilation. And that turned out to help sort our data quite a bit. And I'm going to show you examples of that now. So we looked at the performance of the animal on the task I mentioned before. He has to listen for pure tone. And if he hears it, he licks and he gets a reward. And we looked at the performance on that task over time. And here's green is hits, red is false alarm. So this animal is false alarming a lot and misses. And then we looked at the pupil dilations during these 2 hours. And you could see that the pupil diameter was quite variable over time, like I've been showing. And here are 2 bouts of walking. The black dashed line is the optimal state for performance of the task. So you can see that the animal was varying around the optimal state. Sometimes he's too low, sometimes he's too high. And that gives you the inverted U. Because when we plot the data according to pupil dilation, what you find is that the hit rate, or the D prime of the mouse, was low when the pupil diameter was low. But when the pupil dilated just before the trial, the animal got much better. But then if the pupil dilated much more, the animal got much worse. So it was just like a Yerkes-Dodson curve. The mouse had to be about 40% dilated to do the task well. And this is just group data to show you that. This is the latency to lick, which is kind of a U shape. The shortest latency is this middle pupil dilation. This is the hit rate I just showed you. That's inverted U. That's the D prime, which is a bit of an inverted U. And this is bias, which is a U here. So that means, let me go back. And also, when the animal walked, they did rather poorly in the task. This is hit rate when they're walking. It's not near optimal. You know, people were talking at the time in the visual field about walking enhancing visual responses quite a lot. But we found that the animals couldn't do this task very well if they were walking. They had to be sitting still, all right? And about the same time that we were, you know, finding these things, that walking was bad and sitting still was good for this task, which is kind of a detection task, I happened to get this dog, and I took her to the vet. She's a Samoyed. And when the vet would walk by the door outside, she would freeze and watch the bottom of the door, all right? So you can see that here. Look. Play. And the dogs are great because they have pinna and they point the pinna exactly what they're paying attention to. So she's just looking around and stuff, and then she hears something and she freezes, doesn't do any motion, just stops breathing actually. She stopped breathing and listen, like that. And it turns out that that is, through many different animal species, they have this kind of freeze and listen type of behavior for detection. The Tobias Donor Lab replicated our work in humans they took our data from mice here, and then they did the same task in college students and found a similar effect on pupil diameter and the ability of the student to do the task. So that was nice. And then we thought, well, let's try vision, because in vision, walking is supposed to, extreme arousal is supposed to make visual responses better in visual cortex. So we designed a visual task in which the mouse, head fixed, would watch kind of a snow-like pattern on the screen, and if there was a grading in the pattern, the animal could lick and get a reward. And the snow-like pattern, the grading pattern would be sometimes very light, very hard to see. So here's an example. Whoops, I'm sorry. Let's go back. Let's play it here. It's a movie, but I don't see how to play it. Okay, so there's this, and and there's obviously a grid. You lick and you get a reward. All right, and it's usually random, but there's a grid there now too, and it's much harder to see. Here's the preliminary data we got. It's actually not preliminary. We have now over 120 sessions in this data. This is the performance on the task for a kind of low level contrast, 20% contrast. This is 5% contrast. This is 100% contrast. 100% contrast the animal, it's an easy task. So you need a moderately difficult task to see state effects. So here's a moderately difficult task, and you see this nice inverted U and the hit rate and the D prime, which is the sensitivity of the animal's ability to do the task and distinguish. All right? So it's very nice. It looks like we get the same inverted U on the visual task as we got in the auditory task. And the optimal state is kind of like mid-arousal for them to do this type of detection task. And it's not separated here, but we found walking actually really disturbs the ability of the animal to do this task. So it makes intuitive sense to me. If you want to detect something, you're not going to run to be able to do it. You're going to stand still and focus. So what's the neural mechanisms behind this inverted U? So we recorded neurons in the auditory. Now I'm going back to auditory task. We recorded neurons in the auditory cortex with multi-unit or whole cell recordings. And we looked at the signal to noise, so to speak, of the neural responses. So we looked at the evoked response to the sound. Here's the sound. And subtracted out the spontaneous activity before the sound. And what we found was that the evoked response got bigger, minus the spontaneous, got bigger with low arousal to moderate arousal, but then got smaller with moderate arousal to high arousal. And walking was especially bad. Here's walking in blue. In the reliability of the spiking did that too. Reliability was the trial-to-trial correlation. How similar is the neural response on one trial to the same stimulus on the previous trial that had that stimulus? And that trial-to-trial reliability went up and then back down again with the state. But remember, these states are going back and forth all the time. It's not like, oh, for 5 minutes the animal's drowsy, and then for 5 minutes he's inattentive, and then for 5 minutes he's stressed. These are second by second variations. And so the cortical gain, so to speak, had this perfect inverted U with arousal again, like a Yerkes-Dodson relationship.
Question?
Yeah. Yeah.
So, in your previous graph, you have shown.
In this one or this one?
Yes.
This one?
So you're talking about spike to spike reliability.
Yeah.
And you're calculating the reliability during the evoke period, right?
Mm-hmm.
Shouldn't it be very highly correlated?
So we take a single trial histogram. This is not single trial, this is average, but a single trial histogram, and then do a cross correlation with that with another single trial histogram. And this is where people talk about the unreliability of the brain. In a waking animal, the cross correlation can be very low. That here's 5%, 0.05 means you gave the same stimulus, but the spiking pattern on one trial and another was very different, And the interesting thing is, well, that's if the animal is drowsy or highly aroused. But if you look at the animal in the proper state, it actually goes up to 20%, which is a 400% change, it's still low, 20%, but it's much better than it was at the others. Yeah, that's an interesting thing about neural activity, is that it varies so much to the same stimulus from one trial to the next. And my point is, the big part of that variation is just the state of the mouse is changing rapidly. So we look at this on the whole cell level, and this is now synaptic activity coming into the neuron. And this is synaptic activity to this sound right here. This is synaptic activity to this sound, synaptic activity to this sound. So we're in auditory cortex, and we're driving these synaptic activity with sounds. And the sounds are a randomly presented mix of 6 different sounds, okay? And here in number 1 is a sound presented, and in number 2 is the same sound, but presented when the animal is highly aroused versus moderately aroused. So this was a moderately aroused mouse with the pupil was middle-sized. Then he walked and he got highly aroused, and then the same sound. And you could see the difference in the responses is rather marked because the spontaneous activity changed a lot before the sound. And that's typical. What happens is that with high arousal, there's a lot of spontaneous activity in many parts of the brain. And so the spontaneous activity, the signal to noise, the evoked response to the spontaneous response goes down significantly. All right? I'm going to skip that and just go to this. This is plotting the optimal state in pupil diameter. This is pupil diameter on this axis now, for many different behaviors and neural responses in both the visual cortex, the medial geniculate here in black, and the optimal zone in arousal for many of these measures was around half aroused, half pupil diameter. So it seems that the neural responses in both the thalamus, the cortex, go together with the behavioral optimal state for the animal. So your brain has to be in this correct zone in order to perform the task well. And at that correct zone, the neural representation of the sound is also the best as we measure it. So we're wondering, what is controlling these rapid variations in state? And there are many different possibilities, but neuromodulatory systems would be the first that come to mind. Acetylcholine and norepinephrine are 2 of the most well-studied and prominent of those modulatory systems, but there are others. Dopamine, histamine, serotonin, to name just 3 others. And then glutamate and GABA are also in modulatory systems, and they can rapidly control the state of the brain. So if you look at, for example, if you look at pupil diameter, what controls pupil diameter? Well, pupil diameter is controlled by 2 sets of muscle, circular muscles that constrict to make it small. That's activated by the parasympathetic nervous system. And then radial muscles, which pull to make the pupil large. And that's activated by sympathetic nervous system. And the actual pupil diameter is a push-pull interaction between these two systems. And one is ultimately noradrenergic and the other is cholinergic. And the pathway for this control of pupil diameter is somewhat known, but there's a hypothesized involvement of the locus coeruleus. And this is purely hypothetical, even though it's in the textbooks, because there hasn't been a single study on it that I'm aware of, that hypothesized that the locus coeruleus could activate the sympathetic pathway and inhibit the parasympathetic pathway, and that caused pupil dilation with arousal. So we looked at the axons. Well, let me say, I got ahead of myself. This was all based on one recording. It's the most quoted one-cell recording I've ever met in my life. This was done by Gary Asser-Jones. He recorded a cell in a monkey in the locus coeruleus that seemed to be correlated with pupil dilation. They didn't say what the correlation was. They just show this recording over a couple hours, I think, of the dilation constriction of the monkey's pupil and the locus coeruleus neurons firing rate. All right, and this ended up getting published in Annual Review of Neuroscience, and now everybody quotes that as saying, well, locus coeruleus and pupil diameter are correlated. In my old days, I studied neuromodulation of pyramidal cells in the cortex, and I showed that norepinephrine and acetylcholine and glutamate can, through identified pathways, block a resting potassium current and depolarize the neurons. So in theory, release of norepinephrine or acetylcholine could be causing those depolarizations with arousal that you see in a mouse on a moment-to-moment basis. Other labs, this is what spurred us to study all of this, was, for example, Carl Peterson's lab. This is from Payman Golshani's lab at UCLA. We're finding, before us, this is 2013, these membrane potential changes that look like arousal. They see slow oscillations when the animal was sitting still, here and here, and then they would see depolarization when the animal was walking. And they did some pharmacology and they found that this seemed to be mediated by norepinephrine. So there was some evidence that norepinephrine might be involved too. So we decided to look at noradrenergic and cholinergic axons in the cortex of the animals while they're behaving. So we found axons. This is just a pencil drawing over an axon. That's an actual picture. And we had GCAMP6 expressed in these axons. You can just inject a virus into the locus coeruleus or into the basal forebrain that's Cre-dependent and get GCAMP6 in these axons. Then you can watch the activity of the axon while you watch the pupil or they're walking. All right? So here's an example in my own lab at Oregon of axons in the cortex. This is sped up in time, but these axons are noradrenergic axons, and that's spontaneous activity of the axon. Every blink there is, a change in calcium in the axon, which is indicative of action potentials going on. This Lindsay Collins is the postdoc doing this work. So if you watch these axons and you compare it to walking, which is in green, pupil, which is in black, and cholinergic axon, which is in orange here, or noradrenergic axon, which is in blue here, you see that these axons are actually rapidly changing their activity all the time. This is in seconds here, so this is 20 seconds. And you see that the axons are rapidly changing their activity, which means that presumably the release of norepinephrine and acetylcholine are rapidly changing in the brain on a second-to-second basis. And part of that rapid variation has to do with the state of the mouse. So if the mouse is aroused, if the pupil is dilated, pupil is highly dilated, the cholinergic axons are very active in orange. And that's because the animal is running, or the animal is running because he's aroused. And when he stops running, the pupil starts constricting, the animal is getting relaxed, the cholinergic activity is coming back down, But then there's these little microarousals. And every one of these microarousals is associated with a little burst of acetylcholine release. And that, remember, is associated with depolarization of cortical cells or change in the state of the hippocampus. This is shown on these microarousals on a little faster time base. And then we take a microarousal, idealized microarousal dilation constriction cycle, and compare it to noradrenergic axon activity on average. And you see the noradrenergic axon precedes it and follows it, and so does the cholinergic axon. But you have to remember the pupil is a smooth muscle, and it's delayed about that much, about 750 milliseconds. Sorry. Let's go to, I'm going to go to this. Okay. So with the mesoscope, we can image axons over large part of the brain simultaneously, and that's where we are right now. Because an interesting question is, how regionally specific is this, or how broad is this? All right? You might imagine that when you pay attention to something, you're activating that cortical area, and the other areas are less activated. So that's what we want to do now. We haven't gotten there yet, but we're trying to train mice to pay attention to vision and ignore hearing. And then on another block, pay attention to hearing and ignore vision, and to see if there's regional specificity to this effects, its arousal effects. So our preliminary data, this is an untrained mouse. This is just sitting there. This is pupil diameter. This is walking. This is whisker movements. This is twitching. And this is purposeful whisking. And this is twitching. And these are 3 noradrenergic axons that are separated by about a millimeter from each other on the cortical surface. And these 3 axons are all kind of, first of all, you can see they all are activated by the bout of whisking. And this would be associated with a large pupil dilation, the noradrenergic axons. So the noradrenergic axons are like much more active when the animal's engaged. Not too surprising. But what was also kind of surprising is that even these little bouts of whisker twitching were associated with bouts of noradrenergic activity. Here's an example of whisker twitching. There's no big whisker movements here, it's just these little ones. If we look at the noradrenergic axons, this one's a little noisy. I expand this part here, so this is about 12 seconds here, 12 seconds. The whiskers are quiet. The noradrenergic axons are kind of quiet. And then he twitches for a second, and the noradrenergic axons go up. And then he stops, and they start going down, and then he twitches, and then they go up. And then he stops, they start going down, and there's some twitching. So there seems to be a pretty good correlation about noradrenergic tone across the brain and these rapid variations in the state of the mouse, in the behavioral state of the mouse. So I got one last slide to say what we're trying to do then is we'd like to come up with a circuit diagram of state control and how it affects behavior. And that circuit diagram would look something like this. We want to know, starting from the neuromodulatory systems, how they're controlling the thalamus and the cortex on a cell type by cell type basis to allow these rapid changes in excitability to occur that allow the cortex to respond well or not well to a stimulus and to generate a response. And I'm hoping down the road, in 10 years or so, we'll be able to give you very precise detail as to the ionic channels involved, the cell types involved, and so on. Sorry. And perhaps even the inputs to these modulatory systems and other modulatory systems that are driving them to control the brain on this moment-to-moment basis. Thank you.
you.